Hi everyone, we're super pleased to have Dr. Mostafa Ronaghi here as our guest today. He is a co-founder and now board of director at GRAIL and is SVP of entrepreneurial development at Illumina, running the Illumina accelerator. Before we can go into his impressive background, I wanted to share 2 minutes on PAIR and how we work with founders. So PAIR is an early stage venture fund based in Palo Alto. We're super hands-on and help early stage startups go from 0, the idea stage, to 0.5, the product love stage. And finally, 1, when our startups reach product market fit. We partner with founders in many ways through our student pitch competitions, our accelerator programs, as well as our seed stage investing ranging from 100K to 3 million. Half of our investments are to student founders and the other half are invested in founders from all different backgrounds. I've had the pleasure of working with an amazing PAIR team made up of 7 investment partners who have founded 8 companies and use their operating experience to help our founders with product and growth mentorship. We've invested in early stage startups that are now worth over $4 billion. We also help portfolio companies with making their first early hires and also fundraise post Series A. Yeah, there's a bit of an echo. Mostafa, is it possible to go back to your other laptop?
Sure.
Sorry. Thanks. Okay, I'm gonna keep going and then we can figure out the technical stuff. So we go on this journey in all different industries: biotech, healthcare, consumer, enterprise, and fintech. 4 of our companies have already reached $1 billion valuations. And here's a page illustrating all of our partnerships with healthcare and biotech companies where we worked with them from day 1. So we have Viz AI, Guardant Health that went IPO, as well as BioAge and many more. We also hold many events to share our network and knowledge with early stage founders. For example, we have an event on US immigration for founders coming up November 4th. We have do's and don'ts of company building with Sequoia's Doug Leone coming to speak, as well as our PAIR pitch night for Biotech, which will be judged by Helmi, who is the CEO and co-founder of Guardant Health. And now back to sharing Mostafa's impressive background. So I think the echo is gone now. Thank you for switching back.
Welcome.
And okay, great. So I'm going to go into your really long, impressive background. So everyone, bear with my reading of his long bio. Dr. Ronaghi is a serial entrepreneur and leader in research and technology development and has a wealth of experience in building several successful startups and life science companies. Mustafa is currently the SVP of entrepreneurial development at Illumina and previously served as SVP and CTO at Illumina. He was also co-founder of GRAIL, a healthcare company focused on detecting cancer early, which was recently acquired by Illumina as well, and currently he serves on GRAIL's board of directors. He's also co-founded Advent Home, a sequencing company, which was also acquired by Illumina, NextBio, a search engine for life science data, also acquired by Illumina, and Parallel Bioscience, acquired by Affymetrix. Dr. Ronaghi was also one of the co-founders of Pyrosequencing AB, which is one of the first sequencing technologies that is now one of the underlying foundations for scalable and cost-effective genome sequencing. With that, love to get started. Thank you so much for being here today.
Thank you for having me. It's a pleasure to be here.
Great. I guess I would like to start out with where it all began when you were a teenager in Iran. What were your career dreams? How did it all start?
Yeah, it's when I was in high school, so I had a lot of interest in the medicine. So I was studying actually a lot of medical books, and so my uncle who saw the interest in me, so he asked me if I could go over to his hospital. He was the president of that hospital, managing. It was a private hospital, and then I went there, and he showed me the life of the doctors there, and we went from one clinic to another clinic and another clinic. And, we visited actually 6 different doctors. And after the hospitals, we went to private clinics, outside of the hospitals, where the doctors actually would work and operated in the morning. So they had their own private practice. And after that, so he told him, I just wanted to make sure that, you are making the right decision. This is, of course, a very exciting field, but you really need to see what kind of life you are imagining for yourself. So these doctors, they actually come to work at 5:30 in the morning. They do the operation, and then after operation, they're going to go to their private practice. They basically, they do an amazing job in our society, but in reality, they have a tough life. Their wives, they wouldn't see the husband, and the kids, they don't see their dads, and that's basically the reality of their life. You really want to make sure that you're making the right decision, because it's a big decision. And on the other hand, of course, if you're interested in medicine, there are so many exciting areas in medicine that you could be part of. Still, you're going to have a big impact in the field, but you're not going to see the patients, on a daily basis. And he asked me specifically if there would be any field that I'm interested in. Yes, I'm very interested in genetics. And that was actually in the first year of my high school. And after that, after some thinking, I decided that this is basically the area that I want to explore. This is a new field. And so I went, right after high school to study basically biomedical chemistry, which gave me a good foundation for genetics. So I did my high school in Iran, and in Iran we had actually a very good program in high school. So instead of 5 days in school, so we went 6 days to school, and one day actually was gaining experience basically somewhere, and I chose the hospital labs. So and I rotated in different different labs, from immunology, from genetics, doing all the pregnancy tests, doing all different kind of, blood testing and cytology. And it was actually a very good experience, in those 4 years and in my high school, because I got very good exposure of what the lab testing is about. And in particular, I was actually very interested in genetics, because it's actually very, very interesting. In Iran, since there were actually quite many consanguineous, marriages, so they, by law, 40 years ago, by law, you had to take a lab test to make sure that, you are not basically getting any specific disease like sickle cell anemia and those kind of diseases. So they had to make sure basically that, the boy and girl that they're getting married, so they are not, carrying the same mutation, so that, the kids would not have that mutation. They did actually genetic tests for a few diseases back then. It was mostly, and also they did, the amniocentesis test, which was quite interesting. They would count all the chromosomes, under microscopes and so on. It was very manual, but it was a very good, educational, program for me. Anyway, so after high school, I decided to leave the country. It was war between Iran and Iraq. I realized that, I don't want to live here. And I went to Sweden, where my brothers used to live. And in Sweden, I studied, of course, Swedish. And I basically decided, since I had passed the cycle for applying for the university, I decided actually to do all 4 years of high school again within a year. that I was waiting for
Wow.
university. So I did that again. So it was very good actually that year as well. So it gave me even a stronger foundation. And I got to know the technical language as well, um, for the subject that I was interested in. So um, after my bachelor degree, I went to Royal Institute of Technology, which is the best technical school in Sweden. Um, and because I was more interested in the engineering part of genetics, and the science of genetics were well established at Karolinska Institute, which is a more medical school, and the technology piece was much more advanced at the technical school, which was the Royal Institute of Technology. So I went there and I started my PhD program on microbial sequencing. And after 1.5 years, I came up with the idea of pyrosequencing, which was a novel way of doing sequencing, as I was very frustrated with the old sequencing technology. It was not, very high throughput and laborious, to do the experiments. So I started working on that technology because you could parallel process the sequencing. So going from 96 samples at a time, so you could envision going to a million sample at a time, and much faster and it's more straightforward sequencing. So I started developing that chemistry single-handedly, and I had a lot of lucky moments, and a lot of moments that I had to show my persistence. And finally, I got the technology working. And so when, there was a big invention, that breakthrough that I had in the technology, I asked, my former professor and the guy that I was working with if they would be interested in starting a company together. And they agreed with me, and we started pyrosequencing. And that was actually the first next generation sequencing technology that came into birth. And so there were, of course, a lot of different groups that they were pursuing this idea. And I was actually giving a talk in US, and so I was approached by the head of NHGRI, and he gave me his card and told Mostafa, you want to apply for this grant because we are interested in these kind of technologies, and we are initiating a request for proposal in this area. I told him, I'm not American, so that's okay. Just apply or find a collaborator who is in US and apply for this grant. Your technology is the best technology that I've seen. So I came to US, went around in US, and I was looking for a collaborator as well. And the company that actually I came across with in that 45-day trip that I had was in San Diego, and they were just startup, and they were just 3 people back then. And I approached them and I told, your technology is good, but the sequencing technology that you all combine with your technology is going to make a revolution. And they loved the idea. And that company was called Illumina back then. 3 co-founders.
Wow.
Back in '98.
Crazy.
So they came to Sweden, the co-founders, Mark Chi and John Stupnagel, actually, they came to Sweden, and we started writing the grant together. And we wrote the grant and we got funding. So I had actually money in my hands, and got a lot of good offers from different universities. My offer at Stanford was not the best, but I really liked the area in the Bay Area. The nature, the weather, and amazing, entrepreneurial, community. I decided actually to come to Stanford to start my own lab. And so at Stanford, you can be actually a principal investigator as long as you can bring your money. So there is a lot of universities, you should be a faculty to do that. But at Stanford, they actually led me to be principal investigator. So
I guess in terms of the Illumina meeting the co-founders and getting the grant, how come you decided not to stay with them? I mean, now you've been working with them for many years.
Because I was actually interested in pursuing a lot of different ideas. I had a few ideas, and I didn't want to work for a company. If I wanted to do that, I would do it, for my own company in Sweden. So I decided basically just to collaborate with them. And after a while, they decided to pursue other ideas, actually. So, and I decided to basically focus on pyrosequencing on chip. And so when I was at Stanford, I actually I had amazing colleagues, and so they were very curious. They were very interested in starting companies, and they knew that I know everything. They thought that I know everything, which was not true. But at least, I gave them confidence, to start new companies. And I had a new idea, and we assembled a team to work on that idea. And we started a company called Parallel Biosciences back in 2001. And so Parallel, actually 2 months before launching the product in end of 2002, we realized that actually Illumina is going to be our competitor, because they wanted to pursue multiplex genotyping. That was why they stopped the collaboration with me back then. And so we had actually a very good technology, superior technology compared to Illumina. We could do 10,000 multiplexing, and Illumina would do 1,500 multiplexing. So it was cheaper with our technology and more accurate. And but Illumina had a phenomenal management, and they executed amazingly well. And every time we published something, so they had something, or we had announcement, they had a counter announcement back then. So I was very impressed, how they are thinking, how deep they know our roadmap, and they have something always in their sleeves. And so they had also breakthrough in their technology, and they became very competitive, in the marketplace. And Affymetrix made an offer, to acquire Parallel to compete with them. So we did that, and then after that, I realized actually we are producing a lot of data, and people, our customers, they cannot analyze the data. So I decided to start a software company, and I did that with a friend of mine who was CEO of a software company already in genetic area. And initially I was interested in acquiring their company, but after going deep, into their technology, realized that, their technology is not, scalable for what we want to do. So I asked him if he can sell the company, and let's start this company together. This is the idea. And he loved the idea. He sold the company to Agilent, and then we started the company together, NextBio. And NextBio was basically first of its kind to take a new approach on how to do the data analysis in biological field. So we did that. It was a bit early for the market, so we couldn't scale the market as fast as we wanted, and it was a bit more difficult to do business, in software as standalone company. So, but the company was doing very well, and so we got an offer from Illumina to acquire the company. I had already joined the company, of course. Illumina at the time when they offered. So basically at Illumina, that software combined with the genomic analysis they were providing, became actually a very good, it would solve some of the problems, for our customers. And before joining Illumina, so I was back in 2007, in 2006, 2007, I had a few people in my lab, including Helmi and Tuki and Amir Ali Talasaz. They are who are at Guardant, funded by you. So with Helmi, and Aliaga actually. So they were very interested in starting a company, and we looked at a lot of different ideas. And at the end, I told Helmi, maybe we can start this company. You know, the sequencing. I know people, and in this area, and we know this technology very well. I think it would be safest to start this company. So as soon as we started the company, a week after, Illumina approached me to become their CTO. And I took the call, and I uh, told, I had a good discussion with Jay Flatley, who was the CEO back then. And I told Jay, unfortunately, I'm conflicted, you know, to join this company, join Illumina, although I'm very flattered and, you know, excited about the opportunity. But Mostafa, was the conflict? I told, yeah, I have NextBio who is in data analysis, and I have, you know, started a sequencing company, and they had just acquired a sequencing company called Solexa. And back then they paid $600 million, you know, for a product that didn't have any revenue. And so he asked me, you know, what's the sequencing technology? I thought, yes, I'm not going after your market. I'm actually trying to democratize, you know, sequencing. I'm after a very low cost device. We are thinking to price it at what, $20,000 kind of device, and they were selling $500,000 device. And it doesn't have the throughput as your device has, but you know, it's going to become available elsewhere. And he became very interested in technology. He thought, can I come and take a look? So sure, come over. And he came over. And initially we talked about partnership, if I would join them as CTO. And after a few discussions, we thought, no, we should have it. And he put an offer in front of me. And I talked to Helmi, you know, this technology could actually flourish better, you know, at Illumina. Shall we sell it or not? And he agreed that we should sell it. It was a few weeks old company, and we sold the company and joined Illumina.
So the theme is that if you started the company, Illumina will buy it.
Yeah, they realized it's cheaper.
Yeah, it's a good
to hire me than instead of buying, you know, the companies that I had. But anyway, so I thought, you know, I'm going to join Illumina because they have amazing management. I learned a bit more about business and how to scale a product as they had done. And so I thought maybe I'm going to stay there for like 2 years. I went to the company and fell in love with the company and amazing management. And I felt home, and I felt that I'm doing big things, you know, and I realized their technology has a lot of potential. You know, combine a few features of my technology, actually terminated my own technology program, and I focused on their technology to scale that. And so back then, when I joined, the cost of sequencing a genome was, in 2008, was $200,000 per genome. And it would take 14 days to sequence one single human genome. Mm-hmm. Actually, no, 14 days to produce 1 G of data, sorry. And we required actually 100 different machines to work in parallel in 14 days to produce a human genome. So I basically wrote a roadmap on reducing the cost of sequencing to a level that we can offer below $1,000 in marketplace. Wow. And so within 5 years, we successfully launched actually $1,000 genome. And we could do one single genome, 2 genomes actually, in 3 days, mm-hmm. and with one single device. And wow. With the new device that we have at Illumina, just to put that in perspective, we can actually sequence 60 genomes in 1.5 days with one single device. Mm-hmm. So
What was the key to coming down from that much, the price point that you were at, to 1,000? Like, was it trying to figure out the right technology? Was it trying to get it through the
No, I
process quicker?
felt that the technology is right. We have to just, speed that up and increase the accuracy. So basically, I defined a 10 branch approach. I calculated if I'm successful on 6 fronts, 6 out of 10, I'm going to achieve, the $10,000 genome. And actually, we exceeded on some of those, parameters that some of those goals that we were working on, and we were running behind on some other goals. But collectively, we were actually way below $1,000 genome.
Mm-hmm.
And it was heavy technology development program. But the good thing was that, when I told Jay Flatley, or the CEO, he loved the idea. I told Jay, I have a team of 17 people as a CTO. And if you want me to do this, this is going to require about 110 people. Mm-hmm. And of course, I had a few lucky moments. So Jay decided to support the program, and we were able to achieve, that significant cost reduction. When you look at actually the field of genomics, is very interesting, and I really need to highlight this, because no other field have been able to reduce the cost of genome by 9 orders of magnitude in 25 years. Mm-hmm. And when you look at, back in 1990, the cost of, sequencing human genome was more than $3 billion. And we reduced that cost to basically $1,000, within 22 years. It's more than $3 billion actually back then. They defined a program, $3 billion to sequence one single human genome. But we managed to reduce the cost of the genome by 9 orders of magnitude. People, they hear this and they see that, okay, we broke the Moore's law and so on, but in reality, look at, what is around you. And if you would reduce the cost by 1 billion-fold, 9 orders of magnitude, so what could happen? You can actually have a synchrotron in your backyard. Or you could have MRI machine in your hand, that doesn't require, 9 orders of magnitude cost reduction, but you could have an MRI at your home, for $1. You could actually launch a shuttle to the space from your backyard. Mm-hmm. So that's basically what it means, The closest industry that actually experienced, 9 orders of magnitude cost reduction was semiconductor technology, and it took them about 50 years.
That's amazing.
Telling that, we have done much better than them, or we were more clever than them. No, that's not true. We were actually taking advantage of all the development that had happened in semiconductor technology. Mm-hmm. It became much easier for us with lower investment to actually have 9 orders of magnitude cost reduction. But it came with a lot of dedication and a lot of, hard work by people in this area to make this happen anyway. And we are here now, and we see that, okay, the genome sequencing is about $500 per genome now in larger scales, and it's going to go to $100 in the next few years. So what does that mean? This is going to revolutionize, the healthcare. This is, the impact is going to be enormous. This is actually one of the big foundational technologies that are opening a lot of, venue, opening, a lot of new ways for us to look at medicine or other fields. So you see that, companies like Guard and GRAIL and these companies, they are doing a phenomenal job, and they have phenomenal growth, but the core underlying technology actually is sequencing. And these things would not happen without sequencing.
We have a question from Pejman himself. If you were
Yeah,
going to start a company today, what problem would you be focusing on?
me,
Yeah.
I, I love biology. I love the complexity of biology, and if I would start something today, I would probably, there are a few fields, that I would be very interested in. One is actually single cell biology. Biology, we have to understand the mechanism behind biological function. And sequencing, in a way, is a microscope device that gives a better resolution and better insight of how the biology, functions. But still, we are at the consensus level. We have to look at, biology at the cellular level or subcellular level. So single cell biology is actually very intriguing. I would be very interested in that. And I really think that that's going to actually open up a new frontiers in the way we are looking at medicine.
Are
Um,
there are there interesting single cell biology companies in the Illumina accelerator today, or even outside that you found interesting?
Yes, there has been one. We have had one company. There are other companies that they use single cell to study biology, but it's, in my opinion, you have to improve the single cell at least by 2 orders of magnitude in terms of throughput and maintaining the cost at the same level. And we should have the ability to look at, multi-omics, look at the proteins, look at the RNA, look at the DNA, look at the methylation profile, look at the metabolomes, and so on at the single cell level. And nobody is able to do that today. We just look at RNA or DNA, and we are not doing that efficiently. So we really need to push the biology to get to single cell, because the level of heterogeneity we are dealing with is actually humongous. And so that would limit our understanding about biology. And the next step after that is to look at, subcellular, which means, look at the spatial configuration of those individual components within a cell. So it's basically, as I mentioned, sequencing is a microscopy that we have today. We can look at things at very high resolution, but we need to put in the spatial context and look at other components within a cell and see how those are working together. So that would be one area. The other area is actually the triangle of food, diet, diet, microbiome, and host genome. I'm really interested in, the interaction of, how the food, the reason that I'm picking up the food is because, that's the number one environmental factor that human body is exposed for, because you eat it every day. And that would actually change the microbiome profile, or predispose a certain profile of microbiome in your gut. There are a lot of leading scientists these days that they believe that 90% of the diseases they originate from gut. These are gut disease, and they have manifestation in every other part of the body, like Parkinson. People, they think that today is not a brain disease. It shows itself in the brain. It's actually a gut disease. The same is true with Alzheimer. Same is true with autism. The same is true with allergy, rising allergy.
Did you know that we have our one of our accelerator companies, Interface Bio, is focused on the microbiome?
Yes, I know.
I'd love to dive into sort of your role leading the Illumina Accelerator program and how we think about entrepreneurship in terms of picking companies, is it like the technology more important, or is it the team more important? I think a lot of potential audience members are founders that are interested in hearing about, from the investor side, like how do you pick companies? How do you see them succeed?
So, when you're picking company, at that early stage, like accelerator stage, the number one important factor is a team. There is no question. I would put 90% of the weight on the team. Mm-hmm. They could propose technology, or they could license the technology, or they could have developed the technology. So I would put probably 5% weight on that, and the other 5% weight is probably on the business model, how they are thinking. But as the company, of course, get more matured, at the Series A, I think you have to demonstrate. Okay, it would differ, in different spaces. If you're in therapeutics, you really want to have a lead compound identified, or you're probably pre-IND, or you have pushed that in the clinical trial. That's basically the breaking point for Series A for therapeutics. If it is in diagnostics, I would say, that you should have demonstrated the test, and you should have started actually showing that, okay, the technology that's developed, it has shown the impact, and I can show it in human or animals, that this is working, and this is going to be the impact of this test, when we launch it. If it is in the software space, I would say I want to have more maturity. I want to have customers, testing the software, before putting money in Series A. I think it's different, but definitely at the accelerator level, when they come, the most important thing is the team, no question.
And I know a lot of founders and the team member, I guess the team are very technical and may not have as much business experience, maybe they're first-time founders. What advice do you have to give to these technical founders that still haven't, worked, maybe focus on research more so than business in the past?
I've seen actually a lot of good business people that they have the ability to dive into technology and learn about that. So they have quite good, analytical background. So they can do it, but that's actually quite rare. I kind of feel that it's much easier to develop the technical founder to understand business than the business founder to understand the technology. Especially if you are in high tech or high biotech kind of a space, you really need to have, to understand technology, you really need to understand the market in depth. You have to have the ability to interact, with the leading scientists to basically form a vision for yourself. And I see that actually the technical founders, it's easier for them to evolve, to become a savvier business person than, the business people. And you see that, the very good high-tech companies, they have CEO who understands technology very well, or they have actually a CEO who is technical founder, or technical person, from Microsoft, from, Google, from, Salesforce, you name it, a lot of companies, Facebook. So they have actually a technical person who has evolved to become a business person. And I really think that, they have vision, they are bold, and, so they make proper investments. So I think, for technical people, definitely, get good mentors, write all the relevant books, in this area. You have to listen to a lot of, people, but at the end, you should not follow any single person. You should synthesize your own recipe, because every single startup has a unique recipe for success. And you cannot, just follow them and copy them and become Microsoft or,
Right. So we have some questions, and I know we haven't really talked about GRAIL. We have so many things to talk about. I guess the question has to do with the GRAIL acquisition process. And so I think we can start out by talking about how you founded GRAIL in the first place, and then talk about the evolution of, their relationship with Illumina and so on.
Yeah. So, we had actually, we started looking at blood as liquid biopsy back in 2010, 2011. Um, and back then, that was Amir Ali's project, who is the co-founder of Guardant. And so we had that research at Illumina, and but we did not have, Illumina didn't have the right leader, to execute upon that idea. And Helmi and Amir Ali started, Guardant. We had actually two other companies started from Illumina on ctDNA as well, circulating tumor DNA for oncology diagnostics. And those were Accuragen and Anchorgen DX, and they went actually to China to explore, that market. And um, then, we did not work, on that idea for a couple of years, and suddenly I was approached by Jay, Mostafa, can we work on this idea? I thought, sure, we can do it, but I want to have your commitment to do something with this. And so we decided to go after one of the most challenging area in cancer, which is screening. Screening required actually significant improvement in detection accuracy, detection limit, because when you have got a tumor that's basically the size of a chickpea and in your body, and you don't know where it is, so you should have the amount of DNA that's shed from that little tumor is actually so little that you really need to have amazing detection sensitivity to pick that up. Anyway, we started developing the technology and address some of the fundamental issues in the technology. And after a few months, we got that working, and we demonstrated that actually we can detect a stage 1, 2, 3, 4 cancer, at Illumina. And then I talked to the CEO of the company, Jay, and we decided to spin this company out because nobody was doing that at that time. And we felt that, it's unethical not to do this because we have a lot of people, a lot of patients, who are not knowing that they have cancer, and suddenly they find that out, that they are in stage 3 or 4, and it's very difficult to save them. So when we look at, the impact of what this could mean if you would have early detection, we realize that actually the best cure for cancer today is actually surgery, but you have to detect it, at early stage. And after stage 3, so it's not just one type of cancer that you're dealing with, it's actually multiple types of cancer that you're dealing with. That's why, when you're treating the cancer with certain drug, you're killing, 6 out of the 7, tumor type that you have in your body. And you kill that, and the cancer, you see, oh, it's very good progress. And suddenly after a few months, it comes back and kills the patient. And that's basically the small tumor that, has become resistant to the drug, and the body is weak. So it grows so fast that, it would kill the patient, and there is no solution for that. So we realized that, okay, if anything, when you look at, that happens in the field of oncology, from all the diagnostics, all the screening, all the, therapeutics, everything that we have done so far from the start of humanity. So it has improved the survival rate of cancer patients by 20%. So all inclusive, so it has improved the survival rate by just 20%. We realized that actually if we detect early in stage 1 and 2, so we can do surgery. And so there were studies that show that the survival rate, of those patients is going to increase to about 67%. So see that one single test, if it is done right, it could have a huge impact. So all collective, work that has been done on oncology. So it has improved the survival rate to 20%, and this single test is going to improve the survival rate to 67%. And we felt that it would be unethical not to do this, to the Of course, it's a good business opportunity. It's great for Illumina, but we felt that we cannot do it internally. We spanned it out because it required a lot of investment, very very large clinical trials, and but we were close to GRAIL, and we started seeing amazing data coming out. And then we talked at Illumina that probably we should bring GRAIL back because back then there was no winner, but now in early screening they have the best data, and it would be great to have it back because we have global channel, and we could make this test available to more people than a single standalone company. And that was why we acquired the company back. Of course, it has a lot of synergy with other businesses that we are doing, but definitely, the main goal was basically to make this available to more patients around the world.
I'm curious, so in this, I don't know if you can reveal the specific acquisition process, but I know GRAIL basically announced to go public, and then a couple of days later, it was announced that Illumina was acquiring GRAIL. Was there something going on there, or
yes,
there was
going on, but the company felt that it's very good opportunity to go public. So if the acquisition, a lot of companies actually, when they file their S1 and they are going public, so a lot of companies, they are acquired in that stage. And so at GRAIL, we decided basically either go public, and if we are in agreement with Illumina, then the acquisition could happen. And Illumina moved fast, and the price and GRAIL accepted that.
We have a question around, so someone asked, they're curious about the people around GRAIL, specifically Bill Gates and Jeff Bezos, who are both investors. How did you go about securing them as investors, and how did they help GRAIL grow, if they helped?
My friend Rick Glasner, who was the former head of National Cancer Institute, I actually encouraged him to join Illumina. And so he had amazing understanding about field of oncology. And so he was, at some point, he was actually heading the Gates Foundation. And so he knew Jeff Bezos as well. So he suggested that we should bring Bill Gates and Jeff Bezos as investor. And he had a phone call, and they just followed his advice and they invested.
And then we have one more question, just to make sure we cover everything. So someone asked, since we're on the topic of cancer, can you talk a little bit about the role you believe Illumina should play in cancer genomics from a software standpoint?
Software, Illumina is going to be focused on what it takes to prop the infrastructure of software analysis. The field of software is so huge in healthcare and so much segmented that we cannot do everything. So, but we have to provide the underlying infrastructure for people that they want to do population genome sequencing, and they want to do it at scale. So I would say I would probably draw the analogy to Tesla. Tesla is more a software company than a car company, in my opinion. But when you get their car, so it comes with the underlying software, which is basically how to do the diagnostics, how to basically collect the data in improving the autopilot and improve the autopilot. So those are foundational things, for driving a car around. They may not, of course, they are now trying to populate apps like Netflix and these kind of things, but that's in collaboration. But they are not going to go and, rewrite Netflix, to run it at Tesla. So I really think that, for us, we have to provide the infrastructure from the LIMS, from data collection all the way on the analysis. And that should be part of our sequencing value chain that we provide to our customers, and our customers, they have been asking us to expand, this workflow and provide the complete workflow to them, as they want to do very large scale, kind of genome studies. And these are basic population genomic programs that Illumina has. Um, and we have been doing that, but we are not going to provide, different type of analysis tool on software or cancer. You want to basically correlate drugs, to certain mutations and so on. That's not part of our roadmap at the moment. But there are companies like Tempest, like Foundation Medicine, Guardant. They are actually specialized in doing these things, and
Right.
we would love to empower all those customers than doing something that we don't know that much about.
And we have one more question about about GRAIL, when people are really interested in the acquisition. It says earlier Illumina has divested off GRAIL and then had to buy back at multiple prices. How easy or difficult was it to convince the acquisition?
It was very easy to speed that out because we didn't have resources to do it internally, and we didn't have the right leader to do that. So the decision was quite easy to spin that out. And if you would ask me today if you would do the same thing, I would say yes, because at the end, it would require the right people to execute on the project, and it was not the core things for Illumina back then. The thing for Illumina was sequencing. But once it's developed, then you look at, okay, what's the synergy that I have with this technology? Of course, you have the global channel, you're dealing with a lot of governments, that they would love to implement this. And you financially model basically how the acquisition price should be set, and can you justify this acquisition price or not, and how long does it take for you to make the acquisition profitable, because you have to do it at some point. So we did those analysis actually internally, and we could justify that price.
Okay, awesome. We could answer all the questions. I think we only have like 3 minutes left, so either someone has questions or we can go into wrapping it up with, do you have any general advice for entrepreneurs in the biotech and life science industry? What's one key takeaway out of all of your amazing experiences? What's something that you want to remind people to take away from this conversation?
I see that a lot of people that they are in larger companies and they want to start a company, half time. That's not going to work. You have to leave your academic job or company job and focus 100%, on basically creating something. And any technology or business opportunity has got a window. Window could be anything between 1 to 5 years, let's say. And if you do not execute on that, then you have missed the opportunity. That's why you really, once you decide, okay, I want to start a company, you don't want to do that, half time or part time. You really want to do 150% of your time on it. And initially it would require a lot of work, and you're the only one. And if you are not dedicated, yourself, you cannot convince other people to join you. So definitely leave your comfortable job and get behind this, because if you don't get behind this, you're going to lose the opportunity, and also you're not going to be able to attract, amazing people to work with you. Second thing, you have to be very generous, bring the right people on board. And that's very critical. You should not nickel and dime. Oh, okay, I'll give you, 0.5% or 1%, and you can join me. If the guy wants 2% and you feel that it's 3% and you feel that, the guy is the right guy, do it. I think, the joy of building a company is much more than, the financial gain of it. Financial gain, money, at some point, it would become irrelevant in your life. You have enough of money to do whatever you want to do. Even if you want to fly private, jet going around the world. So after 100 million, actually it would become irrelevant, whether, okay, the jet could be 737, could G5, or it could be, Pilatus, kind of airplane, but you can achieve, what you want actually in your life, after gaining certain amount of money. It depends on, the quality of life, on the standard of life, anybody would have. So I really think that, being generous is extremely critical. You really want to bring, um, good people to help you, and you have to be transparent. Transparency is a huge thing. So there shouldn't be any hidden agenda with anyone, and include everyone on every single aspect of the company, in terms of communication, especially, when you're startup, in early stages, you have to keep everybody in the loop. So it takes a bit more time to do that, but you have to do it. Otherwise, people, they are not gonna be passionate and to be part of your journey. And number four thing is actually, once you have basically um, started a company and you have been trying to pursue different ideas, you see that there are a lot, especially if you have a technology, if you're a technology company, you see that there are a lot of opportunities for your technologies. You have to focus on one, just one single opportunity. Okay, yes, I understand your technology has a lot of potential, but I don't want to see it now. I want that you would focus on this market and show me that you can actually deliver a fantastic customer experience. At the end, customer experience is the most important thing in a company, and that's the energy that customer would bring into your company that's going to warrant, your future success.
Great, thank you so much. I think we're just at time. Really appreciate everyone on the line, and really appreciate Mostafa for taking the time to share your amazing life journey with us. If anyone has any specific questions, feel free to email us at Pair, and we can follow up with anything else there. But yeah, that pretty much wraps up our event today.
Thank you for having me, and you are doing a great job, and I'm really excited